and I'm going to be discussing with all of you today the structural characterization of a gel-forming desiccation tolerance tardigrade protein. So the way that I'm going to structure this talk is first I'm going to talk about tardigrades and their proteins, and then I'm going to talk about hydrogels, which are what these proteins form, and then the aerogels that we're able to create from those hydrogels. So let's get started with tardigrades. And before I really get into tardigrades, I need to tell us why do we care about tardigrades? So in order to do that, I ask another question. How do we get protein-based drugs? Protein-based drugs have become ubiquitous throughout medicine in the past 20 years. From insulin to vaccines, we need more and more of them every year. But when we transport them, we have to manufacture them in a lab, put them to a distribution center. From that distribution center, it goes into houses, hospitals, and all over the world. And in every step of that chain, we need to keep it cold. In 2019, over $35 billion worth of pharmaceuticals were degraded simply due to breaks in this cold chain. And so we have to develop new techniques. So how do we protect our protein-based drugs? So as I've already stated, we use refrigeration. But there's another technique known as lyophilization, where you freeze dry your protein in order to keep it from unfolding, because it doesn't have enough energy in a solid state to change from a natural to a denatured form. But unfortunately, lyophilization can damage proteins. So the technique that you're hoping to use might actually cause more harm than good. Luckily, we have a set of different excipients that you can mix in with your protein to help protect it during this lyophilization. So here's 3 different ones. We have trehalose, which is a sugar, glutamine, which is an amino acid, and human serum albumin, which is a full protein. All 3 of those have already been approved. But when we're trying to use these during our formulations, they each need to be tested empirically. And the stabilization for one protein might be completely different than another. So you could use glutamine and it might work beautifully for this one drug, but then you try another one and it just destroys it. So we always are hunting for more and better excipients. So where do we search for solutions? If we're making protein-based drugs, we started with biology, might as well go right back. And that is where we came from, tardigrades. So tardigrades are extremotolerant microorganisms that have a bevy of fascinating properties. And a lot of them come from the fact that it's capable of being completely dehydrated, entering a cryptobiotic state where its metabolism is completely stalled. And it can stay there for over 10 years. And in this state, it can survive extreme pressures, gamma radiation, extreme temperatures. We even show that if you stick it in the back of a fridge for 30 plus years, and send a graduate student to go cleaning, they'll come back to life. But these cute little guys right here, how exactly are they able to pull this off? How exactly are they able to survive this desiccation tolerance? And that comes down to the ton state. So on the left here, we have a tardigrade. And when you remove all of the water from this tardigrade, it enters the ton state, or barrel in German. And fantastically, once it's in this ton state, you can add just a little bit of water right back, and that tardigrade will be all over the place, eating moss, just like it was before. So this then led to the question of how is a tardigrade able to do this? What is allowing this ton state to give the protection against desiccation? What sort of magical molecules exist here? And a large number of different scientists from all over the world have worked on this, and we've identified a family of proteins called CAHS proteins, or cytosolically abundant heat-soluble proteins, that are both sufficient and necessary for tardigrade desiccation tolerance. This means that if I were to prevent a tardigrade from expressing these proteins and then dehydrated it, the tardigrade would die. But if I were to express these proteins in another organism, such as E. coli or yeast, it will confer desiccation tolerance. It means that you can dry those out and they'll come back to life. Even better, for our first topic again, we have found that they are capable of protecting enzymes. You just mix them together in vitro with some enzymes and it works fantastically as an excipient, which was some wonderful work by a previous grad student, Pixie, and a postdoc in our lab. So in order to kind of study this protein more, we turned to simulation. So this is an all-atom Monte Carlo simulation that was run by Alex Holhouse and Rohit Papu at Washington University in St. Louis. And as you can see here, there is a lot of stuff going on. It's not just one single state. It's bouncing around all over the place. There seems to be a propensity for the internal region to be extended and the end regions to be a bit more collapsed, but even that's not very consistent from frame to frame, meaning that this is an intrinsically disordered protein. So that might not be a term a lot of people have heard, so I'm just going to go over this really quick. An intrinsically disordered protein is a protein that doesn't have persistent structure. So most proteins that you see from day to day or hear about are highly structured proteins. And that means that they fold into a single state. However, a disordered protein samples a large number of states. So if you think about it in terms of the number of possible conformations on this little x-axis right here, a structured protein will have a single peak. It'll only choose one conformation that it'll be in. But a disordered protein will sample a large number of different possible conformations. And this means that they have the ability to interact with a large number of different partners, as well as the environment itself, being sensitive to salts and pH, like unstructured protein might have more difficulty with. So in order to get my research started, I did a primary structural analysis. Unfortunately, CAHS D doesn't have a lot of homology with the rest of the animal kingdom or life in general, meaning that this protein is very unique. This family of proteins specifically is very unique. It doesn't have a lot of matches with other proteins. And as a lot of structural analysis relies on that, it can be a bit tricky. However, this technique uses a variety of different methods, from entropy scoring to multiple threading in order to preserve that information. So this is a representative CHS protein, CHS D, which is what I'm going to be working on through this entire talk. And on this line right here is the primary structure. So this is each of the amino acids that you see in the protein from 1 all the way up to 227. And on the first graph here, you see the percentage of disorder. So if it is all the way maxed out, that means that it's highly likely to be very disordered or sample a large number of conformations stochastically. Meanwhile, the top one is showing the propensity towards different structures. So I've labeled red here as alpha helix, blue as a beta sheet, and the gray as other, meaning that it could be a turn or it could be some sort of hairpin. There's all sorts of possibilities. And what I'd first like to draw attention to is this region right here that has a very, very high propensity towards alpha helices and a relatively low amount of disorder. This was assigned by Martin Blackledge's group as to being an alpha helix via NMR. So all that right there has been already assigned. Fantastic. That's matching our primary structural analysis, giving us a little bit of confidence in what we're doing. And importantly, for my own analysis and for the rest of the talk, there is these more disordered regions where you have a little bit of structure, but it's more likely to have a propensity to sample a large number of conformations. And on top of that, it switches from being fully towards alpha helices to more beta sheet-like structures throughout. And the reason why a highly disordered region can still have a structure is because it is only a propensity towards a structure. That means that if it's forming a structure, it's likely going to be a beta sheet, even though it's sampling a large number of conformations. And with all this in mind, we come to a model of the monomer as a long alpha helix in the middle with some wiggly woggly bits at the end that just go all around and they want to form beta sheets. So now that we kind of got the knowledge of what the protein that I'm studying is and where they came from, I'm going to talk about what happens when you try and purify them, also known as the hydrogel that we get. So to start off, I'm going to show you, this is my model of gelation I put together through all of my work, where we go from a monomer to an oligomer to a fiber, all the way to the junctions that create the gel itself. So we already know how we got the monomer, but let's try and figure out how we can get the other parts of this. So we first identified this gel through, this gel was first identified when, in our lab, when a grad student lysed up some cells. So you make the cells express the protein that we're going to harvest, and then you sonicate them, which makes them all burst open. And then we dyed it with a little bit of blue just to make it look jazzy and pretty. And you flip it over, and that's not going anywhere. CHS D, that's a gel. Alpha-synuclein, which is a control intrinsically disordered protein that we tried, immediately comes all the way out. It's still a liquid. And when you purify it, as you can see here, this forms a really nice gel. This is something that I casted at the bottom of a Eppendorf tube and then stuck on the head of a needle. So that's really cool. What's going on here, we might ask. So in order to study these transitions where it melts and it gels, I need to use a technique that's capable of studying something that in a liquid and a gelled state in a continuous fashion. FTIR has been used to study proteins before, but transmission, unfortunately, has trouble studying a lot of solid samples or studying the transitions of samples, and instead is more favorable towards things such as liquids. But ATR takes advantage of something known as an evanescent wave. So when light is completely reflected by a crystal, when it has a total internal reflectance, it produces an evanescent wave that captures some of the information about the sample on top of that crystal, and then transmits it to the detector, which allows us to see something that might be too opaque to normally shoot light through. So what exactly are we seeing with FTIR? This is the amide region of our FTIR spectrum. And this you can kind of think of as the fingerprint of a protein structure. So FTIR, or infrared spectroscopy, studies molecular vibrations. And the backbone of a protein contains some very nice vibrations for identifying how it's working. And this is through the hydrogen bonds. So Pauling posited that secondary structures of proteins are all formed via hydrogen bond networks. And FTIR studies hydrogen bonds. Therefore, we can use FTIR to study the secondary structural elements. So the amide 1, particularly right here, uses the carboxyl group and the amide on the proton in order to determine, or in order to calculate what the sort of structure we're getting. So if those two are directly in line with each other, then it means it's going to have a very low energy. It's going to be a very happy bond. But as that angle changes, it's going to change the energy of the bond itself, meaning that you'll get a different response in this zone. The amide 2 also contains this sort of information because there's a lot of different vibrations in even simple molecules. But this vibration only really relies on the amide proton. So instead of capturing the interactions between a proton and a carboxyl, you're capturing anything that's interacting with that amide proton, meaning that you get backbone-backbone interactions, but also backbone solvent or backbone small molecule, which means that it's used much less frequently by infrared spectroscopists when studying proteins. So a technique that I could use in order to observe what sort of transition I'm seeing is something called a difference spectrum. So here I have two different states, states A and states B. So if in the terms of my protein, you could think of state A as like being a gel and state B as being a full liquid after being melted. And you subtract state B from state A, and you get a different spectrum. So here, instead of a regular absorbance, I have delta absorbance. Same wave numbers on the bottom, but just a delta absorbance. And anything that's above this line is more common in state A than in state B. Meanwhile, anything below that zero line is going to be more prevalent in state B instead of state A. So with this in mind, we can look at my CAHS D. So we took this is a 50 gram per liter sample of CAHS D that I melted on an ATR. And I subtracted from all of these the highest temperature, in this case, 45 degrees Celsius. And when I subtracted it from all of them, you can see that the blue over here, which is the lowest temperature, about 30 degrees Celsius, has a sharp peak over here, but the melted portion has more of this character over here. And fantastically, we see something called an isospestic point. So an isospestic point is indicative of a two-state transition. It means that we're going from one state to another. We're not seeing a bunch of sub-transitions. We're not seeing it going from a gel to a pseudo-gel to a kind of like a gel, but maybe not a gel, to like a liquid adjacent to a liquid. It's going gel to a liquid. Fantastic. So in order to kind of confirm this, I used a technique called principal component analysis. And I think this is a really cool technique, but kind of going over all of this in my talk might be a bit difficult. So if you have any questions later, I'd love to talk about it. But for now, you can just kind of think of it as me taking each of these spectra, right? Each of these spectra right here. So I just take one of these spectrums, one spectrum, and I take the cross product with a correlation plot that I generated. And this generates a single point for every single spectrum. And I can map that from each of the temperatures and then fit it to a two-state transition. And voila, it works. I got the two-state transition and I was able to characterize it. I used a more classical technique in FTIR spectroscopy for monitoring transitions, where you just pick wave numbers that have the biggest change from state 1 to state 2, and then just monitor the absorbance at each point. And I compared it against the results from the principal component analysis, and it outperformed all of them. So I managed to beat my own bias there, which was quite gratifying. I found that our protein can melt around 37 degrees Celsius, and that it is the concentration of our gel dictates the point at which it melts. It has a positive delta HM, and that delta H of melting is on par with the same sort that we'd expect from a globular protein of a similar size, meaning that the density of hydrogen bonds that are being broken during melting is similar to a protein unfolding. Fantastic. So everything's kind of sort of making sense. This principal component analysis is a valid method of studying this. Now it's time to actually study the structures and see how they change when I melt this protein. So as I mentioned before, the location in this amide 1 dictates what sort of structures you get. So here are 3 different structures. We have the random coil, the alpha helix, and the beta sheet. So the random coil is centered around 1645 generally, and it has a very broad distribution. This makes sense if we think about the angle argument again, because those random coils, it doesn't really matter where they're at. They're just going to be everywhere, and they're just going to have a very large standard deviation. But the alpha helix, that needs to be much more canonical. If your angles are too far off in an alpha helix, it just breaks. It's no longer an alpha helix anymore. So any alpha helices you see are going to have a very sharp peak, and that's going to be centered around 1650, 1652. Beta sheets, on the other hand, have 2 separate responses, one at 1630 and one at 1680. And there's a lot of different things that are going on inside these peaks, but for the purpose of this talk, I want you all to think about 1630 as being more related to intermolecular beta sheets, so beta sheets happening between 2 different chains, and 1680 being more related to intramolecular beta sheets, or beta sheets that are forming in a protein. Okay? And okay, you think, fantastic, we now know this. I can just take my amide 1 and just slot all these in, and good. Unfortunately, it's not really that simple. So here's an example that I found on the internet of some people trying to fit their data. And you can see it's a bit of a mess with all the stuff that they have to try and figure out. They have 2 different alpha helices, one of them centered around 1660. They have a couple different beta sheet responses over here. They're convolved with loops and turns. It's very difficult to identify exactly what you're seeing sometimes. And so in order to try and get around this, the first thing that I did was I took my amide 1 and I got the second derivative of my spectrum. So what that means is if you took physics, remember that, we're going from a position to an acceleration. So it's the change in my graph. And any of the little peaks that I see here can be generally correspond to different structures I might see that are changing or are present inside of my protein. So I can mark these out, and you can see on the right-hand side, they're pretty obvious, but the left-hand side, I'd be hard-pressed to try and find that myself, I'm not gonna lie. So now I found that there's a beautiful peak in all these different locations. I can fit those, but one major problem kind of popped out for me, which is there's a peak at about 1650, 1648, and that is right between the random coil and the alpha helix, and they convolve pretty nastily. And so how do I know which one is actually changing during my gelation event? Could it be forming strange random coils all over the place during gelation? What might be happening? This is where I return to the PCA. So remember how I showed that plot that was made with my PCA? This is the amide 1 again, where you have that low and that high frequency. It goes right through that isospecific point. I caught that transition, fantastic. But what is really nice about this is it also caught the amide 2. So I can see the amide 2 dependence on gelation. And so this peak right here is very much upregulated during that transition. And I can assign that as an alpha helix, because despite the fact that the amide 2 is not frequently used by protein spectroscopists, it is still capable of being assigned. It's just very difficult to disentangle. And this alpha helix is not convolved with a random coil. meaning that I'm able to assign the peaks that I found in my previous analysis at 1650 to an alpha helix during gelation. So great, I now know what my change is. This here is a graph with the temperature on the x-axis. We're going from a low temperature to a high temperature. You can think of this side as being the gel and this side as being the liquid. And as we increase our temperature, we're seeing the alpha helix drop significantly and the intermolecular beta sheets drop significantly. Okay, so those two are involved in our gelation process. And we see a rise in these turns, these intramolecular beta sheets. That means that we're having a transition from these two structures into these two structures. And I did some more research because a day in the library saves you a month in the lab. And I found an interesting feature that can pop up, which is if you have an alpha helical resonance at about 1650 and an intermolecular beta sheet resonance at around 1630, this is indicative of a coiled-coiled interaction. So this implies that the next stage of our model of gelation goes from a monomer to a coiled-coiled oligomer with still some sticky ends sticking out of there. And now, in order to try to go deeper, I would turn to rheology. So I'm guessing a lot of y'all probably haven't seen much rheology before, but you can think of it as the study of flow or the study of how solid or liquid-like an object is. And you do this by putting one plate on top of another, and then you wiggle one of the plates, and you have your sample transmitting that wiggle to the other plate, and you measure how much of that wiggle gets transmitted. So in the case of a perfect solid, which is right here, you have the strain, which is the input wiggle or the input force, and you have the stress, which is what is detected by the instrument. And in a pure solid, those are going to be perfectly in phase, which makes sense. If you put two solid things together, they're just going to wiggle together. It's going to transmit it perfectly. Fantastic. And in the terms of a liquid, these are going to be out of phase because it's no longer coupled directly. And so this allows us to determine the strength and the energy, how much energy a sample can store and how much energy it is able to disperse. We call these the storage modulus and the loss modulus respectively. So here I have 3 different concentrations of CAHS D at 30 grams per liter, 40 grams per liter, and 50 grams per liter. And the 30 grams per liter melts at a much lower temperature than the 40 and the 50, as you can see, because it starts at 293 and it melts at around 306, 305 Kelvin. But the 40 grams per liter sees a massive increase in both strength and melting temperature. So here it goes from about 200 pascals all the way up to 1,000. And from 40 to 50, it goes up by 1,000 again, so up to 2,000. And in all of these, despite melting, we see that the storage modulus is always higher than the loss modulus, implying that we have a persistent structure even after melting our samples. But I know that most of y'all haven't been spending a lot of time trying to translate the idea of a storage modulus to a different strength. So I'm going to use something else that everyone should definitely be aware of, which is the strength of different human tissues. So we take that 20 gram per liter. It's about the difference. It's about like halfway between some like snot that you had this morning and your brain tissue. And if you look at 30 grams per liter, it's like, between your lung and your brain. And if you do like 40 gram per liter, that's like kind of like between your lung and your liver. And 50 gram per liter, it's like your liver. Okay, I get it. Most of y'all didn't take zoology and had to cut things open and poke the insides for a grade. But I'll give you another thing to completely ruin any desserts you have with this. Jello has the exact same strength as our 50 gram per liter CAHS D, which has the same strength as the liver. So next time you eat some Jello, think of a liver. But fantastically, we got this thing. It can change what temperature it melts at, and we can change how strong it is. And those two properties together make it a really good candidate for being used in biomedical applications. Because while it might be a little weird that we can tell what sort of tissues it's like, it's also useful because if you try and implant a hydrogel into a person, maybe that's one that's impregnated with a protein that you're trying to deliver, if it's not the right strength, the body's going to get very angry and it won't heal properly. So going back though, we want to look at we're trying to understand what sort of structures we're seeing. And with this, I used a microstructure. I can use the different mechanical properties of our sample to identify the different possible microstructures it can hold. And so here is once again this picture of a gel I had. And this time I want to draw attention to the fact that this gel is holding its shape on the head of a pin. And there is a little tear at the top. It is not shifting or melting over time. It's not slowly falling down. And I used a technique called mechanical spectroscopy or an angular frequency scan. So remember how I was talking about the wiggles? Well, if you change the rate at which one of the plates wiggles, it changes the response of the material. So I have family from California here today, and they're probably very aware. If the earth wiggles in the wrong way, it can turn into a liquid. So in the same way, if you wiggle a sample properly, it'll eventually turn into a liquid. However, under the frequency regime that I studied, which is a fairly large one, the storage modulus is independent of the angular frequency. This is a hallmark of a strong gel. And we have a cold setting, strong gel that is clear in character. And this, according to the literature, is a very clear sign that we're dealing with a fine-stranded, flexible fiber as our microstructure. So fantastic. We found that our next step in our gelation cycle is that we're making fine-stranded fibrils that are very flexible. Okay, we got the next step. We're doing great. But how do we figure out how these fibers come together? Because right now, all we know is that there's a bunch of fibers in there. We know that they're made out of coiled coils, but how do they stick together? So with this, I turned to a constant temperature experiment. So in this one, instead of taking one concentration and melting it, I took a bunch of different concentrations and studied them at the same temperature. So in this case, we're going from 20 to 50 grams per liter, which is going from a liquid-like state to more gel-like state. And the first thing to note is that there is essentially no change in the composition of alpha helices or turns in this sample. However, what we do see is that during gelation, there is a rise in the intermolecular beta sheets and a drop in the intramolecular beta sheets. And if I can call everyone's minds back to that long ago, back to that primary structure analysis, there are very sticky N-terminal regions that are very disordered and have a propensity to form beta sheets. So what this implies is that we're forming junctions with these beta sheets where the strands are coming together and they're able to interact with those sticky N-terminal strands, and they're transitioning from intramolecular beta sheets, where they're interacting with themselves, to intermolecular beta sheets that are holding the gel together. Fantastic. I've gotten a really nice model for how this thing gels. I'm feeling pretty comfortable with this hydrogel. I've got some good mechanical characteristics. I think it's time to turn to the next way we can look at this, which is an aerogel. So in order to kind of talk about an aerogel, I have to talk about how I made it. So I created these aerogels through a technique called lyophilization, which I mentioned very early in this talk. And this is relevant as well because this is how we're trying to store our proteins. So in regular evaporation, you just put a sample, in this case like a protein, into some water, and you evaporate it. And during this time, there's a lot of energy, there's a lot of space and time for the protein to unfold because it's not going to like being evaporated. And in that case, you ruined your protein. It no longer functions. However, in lyophilization, you flash freeze your protein, lowering the energy density of the sample, and then sublimate off the water using a vacuum. And in this way, you prevent the protein from having the energy to really unfold and help protect it against, well, lyophilization-induced or evaporation-induced damage. Of course, as we talked about earlier, this isn't a perfect technique, but our proteins are very well suited for it because they're designed to be dry out. So for the rest of this talk, I'm just going to just say this right now. When I say concentration, I'm referring to pre-lyophilization concentration because it's kind of hard to get a concentration from a dry solid. So the technique that I used to initially study these was scanning electron microscopy. So this is a tardigrade that was taken by the Goldstein lab, or an SEM image of a tardigrade taken by the Goldstein lab. And this is kind of how an SEM works. You shoot some electrons at a sample, you bounce it off and read it in a detector, just like you would with regular light. But because it's an electron, you can see things much smaller than normal visible light would usually allow you to. And this is kind of the results that I get. So it's kind of overwhelming to look at first. So let's just go one by one. I have a 0.1 gram per liter sample here. This means that I had a sample that was at 0.1 gram per liter and then lyophilized it. And this is a scale bar of 50 microns, and this is the same scale bar for all of these. And we have a 5 gram per liter sample here and a 40 gram per liter sample here. And then I also have two little bars up here, this blue bar and this red bar. The blue bar corresponds to, the blue and red bars correspond to 50,000 CAHS D molecules laid next to each other, put end to end. And the blue bar is if you assume that the CAHS D is collapsed like a globular protein would be, and the red is if it's fully denatured or extended. And so that kind of gives you a scale for how large these structures are in reference to a single protein. And if you look at the 0.1 gram per liter, it's a mass of tangles. There's a lot of very fine strands throughout it, and there's all sorts of things going on. At 5 grams per liter, you're starting to see more and more structures appearing, with the strands thickening up and these more slab-like structures appearing. And at 40 grams per liter, you see the pore structure of the actual gel appear. And so for the rest of the talk, the two features I'd really like to focus on here are the fibers that we see and the slabs that we see. So the thing that kind of jumped out when I did the aerogel SEM analysis was, how do we go from these singular junctions all the way to these massive slabs all lined up next to each other, forming the walls of a pore? And that was kind of a tricky thought. So I started looking actually at the fibers. So I started taking the diameters of the fibers using image J. And you can see as you go from 0.1 gram per liter up to 10 grams per liter, you see that it's this pretty linear increase in the average. But the distribution gets wider and wider, where it's going larger and larger and smaller for all of them. But it's still increasing, right? That's fine. Except when you get to 40 grams per liter, it's not. 40 grams per liter, it's significantly less than 10 grams per liter. So in order to kind of look at this data a little bit deeper, I got some kernel distributions and fit them to the data that I gathered. And at 0.1 gram per liter, you see that a really sharp peak that appears at around 0.15 microns. But then once you go up to 0.6 and 1 gram per liter, you start seeing two more peaks forming. They're a little bit different, but you see little shoulders right where the peak of the 0.6 gram per liter is on the one, and vice versa. You go up to 5 and 10, and it gets messy. It's all over the place. It's sampling all these distributions. It's getting bigger and blobbier. But then you go back to 40, and it is just right on top of that 0.6 gram per liter and that 1 gram per liter. And there is nothing past it. So what I am hypothesizing is that after a certain thickness of fiber is reached, it transitions into another structure, such as the slab. And if you're below a certain concentration where it's allowing the sample to gel, it doesn't have the opportunity to join those slabs, and that's why we see our thickening in the medium concentration samples, but none of it in the highest concentration sample. So in this case, what we'd be seeing is we have a fiber, and that fiber starts thickening and becomes a slab. And so in order to figure out how these slabs kind of came together, I wanted to look at the difference between the aerogel structures in FTIR and the hydrogel structures. So here is a graph that's the delta secondary structure. So it's the change in the percent of secondary structure between the aerogel and the hydrogel. If it's above this axis, the aerogel has more of this secondary structural element. If it's below the x-axis, the hydrogel has more of this structure. And what I want to call attention to is that the intermolecular beta sheets that we see here, those collapse to the same point essentially. The aerogel seems to have less of them at the lower concentrations, but at the higher concentrations, it seems to collapse and they both reach unity, where they're essentially the same. But instead of having that transition be exactly identical to what we'd expect from the intermolecular beta sheet, we're seeing a massive increase in the aerogel to the intermolecular beta sheet, instead of going back towards unity like we would have expected if the aerogel and hydrogel were achieving the same exact result. And the alpha helices in turn are having the same sort of decrease. So what I'm hypothesizing is that the formations of these slabs are resulting in a large number of these coiled-coiled helix interactions, and that they are themselves coming together and allowing for a larger number of big beta sheets to form. So the beta sheets that I talked about in the hydrogel, those were fit at 1630, and those were relatively small if we consider it. They should be between 3 to 4 strands apiece. But the ones that we see in the aerogel tend to be much, much larger. So what I'd hypothesize is that as these slabs start coming together, they kind of zip up on the ends, forming a larger beta sheet across as they interact, sticking together the walls of these pores and forming the final aerogel that we saw. So taking together this dry data and the wet data, the full model of gelation, aerogels and hydrogels together should be that we have a monomer with 2 sticky ends and an alpha helical portion in the center that becomes a coiled-coil helix with these sticky ends once more, and that forms a thin, flexible, fine strand that then thickens up, allowing the formation of slabs, which can form the walls of pores, and those walls of the pores then stick together using the intermolecular beta sheets. And I'm pretty happy with all that. I feel pretty good about getting all this stuff done, but I recognize that this is all being done in vitro. I am looking at samples in a test tube, not in a cell. So I did one last experiment where I took those cells that we saw way, way back when we first saw these proteins, the lysed cells. You take some cells, make them express a protein, and blow them up, and then look at what's inside of them. And I lyophilized them. So this one first is what an empty vector looks like. So this is a 1 millimeter scale bar, and if I just take a random cell, don't have it produce anything in particular besides kanamycin resistance, and blow it up, it looks like rock climbing. It'd be great for bouldering if you're into that. But if I make it express CAHS D and lyophilize it, a large number of structures start appearing, and it's very ordered. So this is 1 millimeter as well. And if we zoom in on this region right here, you can see that this looks very similar to that in vitro gel that we've been looking at for the past 15 minutes. But what I find fascinating is that there is another structure that also appears, which is highly ordered, and I frankly don't quite understand how it works yet. But it is very telling that when we have the presence of biological substances in our gels, it can radically alter the structure of the gel itself, pointing to a very large and beautiful field of study for future people to look at. So with that in mind, let's talk about the future directions. So I'd say that one of the first things we should try is just a bunch of different drying methods. So I brought up lyophilization, but there's a lot of different ways we can dry. You can heat things up. You can use a Genevac to spin things down while evaporating. And there's a bevy of proprietary pharmaceutical methods that would all benefit from that we could benefit from by looking at our protein with. Using things such as liquid observed vapor exchange experiments or hydrogen deuterium exchange. This will let us understand what sort of residues in a protein are exposed in the dry state to the vapor in the air. You can do this with both NMR and FTIR if you so please. We can use cross-linking mass spectrometry, both in vitro and in vivo, to see what parts of the proteins are interacting with each other and try and parse out what is relevant or important for protecting a protein. We can expand the cell lysate lyophilization experiments with FTIR and SEM so that we can kind of get a better grasp on what that strange structure we saw that was interacting with other biological molecules. And we can use other CHS proteins. There's a variety of them. This is a family. I studied a representative one, and it's a pretty good one. But CHSG is twice the size. CHSA, S, and Q have different sequences that mean that they have different charge properties or different PIs. And all of these are promising things to look into. And with that, I'd like to thank my lab. I'd like to thank everyone. I'd like to thank Candace for helping get this project really going and getting me set up in the lab when I first joined. I'd like to thank Pixie for all her work getting this project into the lab to start with. Oscar for taking over the work on this after I leave. Harrison for the lovely work he did in helping purify this protein to make sure we had enough of it to actually work with. Rinko, who's an undergrad who helped work with some of this. Julia for being my contemporary that I could talk about all this stuff with and It was fantastic. She even worked on some of my papers too. We worked together. And I'd really like to thank the Atkin Lab specifically for helping with the FTIR stuff. Y'all saved my bacon a couple of times on some difficult stuff. And I'd like to thank the Shaco Labs, particularly Ben and Fouad, who helped me with my rheology, and Wilton and Amar for helping me with different controls and experiments that I've run, Amar for the SEM, and Juanita and Rita for helping me get the instrument up and running so I could actually gather data, Rafael for helping me automate my instrument so I could more easily gather data, and Luke for wonderful work he did on the animations. And I would like to thank my funding sources, the NIH and Orange Data Mining, for making a program that I could use for free to do a lot of analysis that would normally cost a lot of money or a lot of time to do. And finally, I'd like to thank my committee for judging my work today.